And the next person up is going to be James. James, brilliant, James. J- James, what is it you're going to be telling us about? Photosynthesis. Okay, I like this. I'll load up your slides. You give me the nod when you're ready to start.
Okay, so hi everyone. Um, I'm a first year PhD student at the University of Liverpool, and I'm working in photosynthesis and carbon fixation. So we're trying to look at ways in which we can try and deal with the climate crisis as such as what it is through photosynthetic means. So, so yeah, so funny enough, these are actually AI generated images of what they think is photosynthesis. So, um, yeah, basically, what my work is looking at is using bacterial photosynthesis rather than looking at plant photosynthesis. And so, the general idea that people get of photosynthesis is that it's plants using sunlight energy to convert carbon dioxide and water into oxygen and sugar. And for the most part, that is what happens. But on Earth, there is a wide plethora of different organisms that can perform photosynthesis, such as bacteria. And so in our lab, we work in different types of bacteria that can perform this different versions of photosynthesis that are very different to what plants can do. So plants use carbon dioxide and water, importantly, to produce oxygen and sugar. And this is known as oxygenic photosynthesis. So they're producing oxygen and they're using light energy to drive this process. But in the bacteria that we work with, they work in strictly in the absence of oxygen, most of them. So they don't produce any oxygen. They'll use hydrogen bound to X, which won't be oxygen, releasing something that isn't oxygen as such. They might use sulfur or something. So what are the structures of these photosynthetic organisms that they actually use to absorb light and convert it into organic matter? And again, these are AI generated images. So the general schematic is that there is a surrounding peripheral sort of antenna complex that is absorbing the light and it's funneling the light energy into the central reaction center. And the reaction center is what abstracts or takes electrons from water or from sulfur or from hydrogen gas and funnels those electrons into central metabolism. And this is all powered by light energy. So if we look at how primordial planet used to look at the beginning, before life really emerged, it was a very volatile place. And so life couldn't exist on terrestrial land as we know it today. But what happened around 2.5 billion years ago, there was a great oxidation event where there was a huge upsurge in oxygen that led to the creation of ozone in our atmosphere, which protected our planet from UV rays. And this was all thanks to cyanobacteria, which are capable of performing this version of photosynthesis, where they crucially release oxygen into the environment. And so without this, we wouldn't be here today, essentially. So thanks to our endeavors as the human race, we've been huge amounts of greenhouse gases into the atmosphere, creating a huge imbalance where photosynthetic carbon fixing organisms can't compete with the rate at which we're releasing this amount of gas. So the goal of my work is to create a novel version of photosynthesis that doesn't exist in nature, but would in theory remove carbon dioxide at a much greater rate than what plants can do and any other photosynthetic life form can do currently. So again, the purpose of the work, the efficient version of photosynthesis that could vacuum up carbon dioxide, carbon monoxide, and different gases much faster than what other organisms can do concurrently. And so if we look at the phylogenetic tree of life, you have the basic schematic, you have bacteria, archaea, and eukaryotes, which is where we're found along with plants and all the other complex life forms. And that's what you're taught at school. It's quite a simplistic idea, but in reality, it's a far more complex structure. And the organism that we handle is hidden up here, the Firmicutes phylum, and it's heliobacteria that we're working with. And they're a form of light absorbing bacterium, but they can't take on carbon from the atmosphere. And so heliobacteria, they use light, but oxygen kills them, and they can't take up carbon dioxide, but they will use sulfur or hydrogen sulfide or sulfuric gas of some kind to power their metabolism. And so what we want to do is put this initial carbon fixing process called the Wood-Lungdal pathway, which plants can't do, and then augment it with further carbon fixing pathways, glycine synthase, methane fixation, RTCA cycle, to create a sort of supercharged carbon fixing organism. But in order to do this, you need to give it enough energy. So you'd have to allow it to absorb more light to power this process. So in order to do that, you'd have to give it additional pigments, things called chlorosomes, pyridine and chlorophyll A, to upscale it. And so then what we'd like to do, if we can get this working at the single cell level, is to put it into, say, bioreactors, where we can put these into really large containers where we can control the gas content, which are like these structures here. And then my inspiration for this really comes from the idea that if you can running, you can then put this possibly beside factories or uh, vehicles that are releasing the carbon at its source and prevent it reaching the atmosphere in the first place. And so we could, yeah. And that was essentially all of it. Thank you.